Hi, my name is Tanylse, and thank you for taking your time to join us today. We're going to talk about a molecular biology technique called polymerase chain reaction, or PCR. So let me quickly show you the aspect that we're going to look at in a little bit more details along this presentation. We're going to talk about the purpose of the technique in the introduction and what PCR is used for. When it was discovered, how this technique was developed, followed by how does PCR work, its application. We're going to see some examples how PCR is applied in science and the advantages of the technique, the shortcomings of the technique, and the last aspect, but not the least, covering this presentation is the potential future applications or extensions for the PCR. Polymerase chain reaction is a method of amplification of DNA. That's it. The DNA is multiplied through a reaction. PCR has revolutionized the field of molecular biology because it's an easy process that allows you to make millions of copies of genes. Simple, it's a DNA duplication method. This technique is a copy mechanism for the genetic material. It's like a molecular photocopying. So, using PCR with a small segment of DNA, which you can call target DNA, it's possible to copy the DNA many times. In simple words, PCR is a rapid amplification of tiny fragments of DNA. Important in this technique is that it can recognize rare DNA sequences into extremely complex mixture of biological materials and also even a whole cell structures. So, once this sequence is identified, that could be from a single human hair or a saliva sample, then it can be amplified through PCR. That's why PCR is very important and highly used biotechnology for molecular biologists. What is PCR used for? Once amplified, the DNA produced by PCR can be used in many different laboratory procedures, such as diagnosis of genetic diseases such as cancers and tumors, detection of virus such as influenza virus or HIV virus, detection of bacteria. It also can be used to match organs donor with recipients in transplant programs, establish paternity. So the PCR has several uses, all very interesting, and the list doesn't stop here. PCR is also used for identify possible suspects whose DNA may correspond to evidence left in the crime scenes. It's used for the study of human evolution, detection of mutation, DNA clone of prehistoric biological materials such as mummies or dinosaur bones and extinct organisms. So its clinical application and laboratory uses is very valuable because PCR is an essential tool for scientists and many researchers that study genetic material. How PCR was discovered? Carrie Muslin was the creator of the PCR in 1983. He was working for one of the first biotechnology companies, and his role was to make short chains of DNA, the DNA synthesis. He stated he was been trying to think about an innovative way to analyze the mutations in DNA. However, he realized instead he has invented a method to amplify any regions of the DNA. The technique was published in 1987, and in 1993, Muslin and his colleague Michael Smith shared the Chemistry Nobel Prize for the PCR invention. 3 years before to receive the Nobel Prize, he wrote in the Scientific American, beginning with a single molecule of the genetic material of DNA, the PCR can generate 100 billion similar molecules in an afternoon. The reaction is easy to execute. It requires no more than a test tube, a few simple reagents, and a source of heat.
Hello, my name is Sama and I'll be going through the procedure of the polymerase chain reaction as well as the current applications of the technique. To begin with, how does the polymerase chain reaction work? Well, firstly, what is needed for PCR to commence? PCR requires a DNA template which has the target sequence, as well as DNA primers, which are short pieces of single-stranded DNA complementary to the target sequence. Next are nucleotides, specifically called deoxynucleotide triphosphates, or for short, DNTPs. And these are the single bases that are the building blocks of the replicated DNA strands. They are adenine, thymine, guanine, and cytosine. For replication to occur, DNA polymerase is also added, which is an enzyme that synthesizes the new DNA strands. Enzymes act as catalysts and increase the rate of reactions. This is important for DNA replication, as it should be extremely fast. The most common DNA polymerase is Taq polymerase, which is from the Thermus aquaticus bacterium. This type of enzyme is thermostable and won't denature when put under high heat conditions. PFU polymerase from the P. furiosus archaea is also another commonly used heat-stable enzyme, due to its high-fidelity proofreading abilities, meaning there are fewer errors compared to Taq polymerase. And last but not least, a buffer is also added to optimize conditions for the reaction. This buffer contains magnesium chloride, which is a cofactor for Taq polymerase, and allows the enzyme to work correctly. So the polymerase chain reaction begins with the mixing of all the components in a test tube, which is then placed inside a thermal cycler machine. PCR consists of 3 steps: denaturation, annealing, and extension phase. This machine allows the 3 stages to be repeated in every cycle by raising and lowering the temperature until an ample amount of DNA has been replicated. The image on the slide shows an overview of the procedure. The first stage, denaturation, requires the double-stranded DNA to separate into 2 single strands by raising the temperature to 94 degrees Celsius for around a minute. This increase in temperature causes the hydrogen bonds between the nucleotide bases to break. As you can see, one strand runs 5' to 3', while the other runs 3' to 5'. Up next is the annealing phase, where the temperature is lowered to around 54 to 60 degrees Celsius. Because the temperature is cooling down, this allows the primers, which are complementary, to attach to the target sequence in the DNA template. The specific temperature is dependent on the sequence and length of the primer used. If the temperature is too low, the primer can bind to other sequences in the template DNA, which will result in errors. However, if the temperature is too high, the primer can fail to bind at all, which will result in 0 or decreased amplification. This step usually takes less than a minute. The last stage of PCR is the extension, or sometimes known as the elongation phase. For this stage to begin, the temperature should be raised again, according to the optimum temperature for the specific DNA polymerase used. Since Taq polymerase is mainly used, today we'll talk about the optimum temperature for Taq polymerase, which is around 72 degrees Celsius. The polymerase produces the complementary DNA strands by adding the nucleotides onto the original strand by about 1,000 base pairs per minute. This takes at least 30 seconds. And so the cycle repeats itself over and over again until there are enough copies generated by the exponential growth. After the 30th cycle, there will be over 1 billion copies of DNA. Now, how is this popular technique used in everyday life amongst scientists and professionals? The current applications of PCR are evident in many fields. In the medical field, PCR is used to detect and identify viruses and microbes that cause infections and diseases such as HIV and AIDS. This is also useful when detecting new strains, providing greater knowledge about the behavior and infection pattern. It is used for detection of genetic defects that are associated with diseases. For example, cancer and sickle cell anemia, along with the susceptibility to these diseases. In dentistry, it can detect dental caries, which are caused by bacteria, as well as oral cancer and many other diseases. PCR in forensics is useful for comparing DNA samples found on scene to help solve crimes. Samples such as hair and bodily fluids, like blood, can be evaluated by amplifying the DNA and matching them against possible suspects, proving them either guilty or innocent. Furthermore, scientific research has benefited greatly from PCR. It can help to understand biochemical and signaling pathways and also drug mechanisms of action. The speed of the technique allows information to be acquired much faster, making research easier and more time efficient. Also, because of its amplification properties, naturally it is used for cloning. And by altering the DNA polymerase used, errors can be reduced. PCR is also handy for recognizing and quantifying the species present in food. This is important for food production, as detection of specific components is important for many reasons, such as health or religious factors. The smallest amount of a specific species can cause an allergic reaction that could be fatal. For example, people that are suffering from celiac disease cannot tolerate even the smallest quantities of gluten. Due to its sensitivity, speed, and simplicity, PCR is ideal for this. Food spoilage can also be spotted by the presence of microbes. PCR is also used for the risk assessment of microbial pathogens and other chemicals present in drinking water, as well as recreational water quality of bodies of water, such as pools and lakes. Of course, these are just a few of the many applications of PCR used globally today.
Polymerase chain reaction advantages. Polymerase chain reaction, or PCR, is a very popular method for duplicating DNA, and for good reason. It is well known for its rapid speed without foregoing accuracy. This speed allows for it to be easily redone multiple times without much effort. From a single gene, PCR can make up to millions of copies of a section of DNA. This only requires a very small amount of genetic material to create more copies. This can also be in many forms, such as skin, hair, and blood, all of which can be readily available and non-invasive. According to Wassenegger, when analyzing and replicating with plant DNA, PCR has increased the speed of many methods and outright rendered others obsolete. Northern and southern blotting for analysis can be effectively replaced by PCR due to needing much more DNA, being more expensive, and needing long preparation times. Its greatest advantage, however, is the fact that foreign DNA can be examined and identified from under 200 picograms of DNA, suggesting great sensitivity. This gives it a distinct advantage over many other forms of amplification and analysis. PCR can be used to detect many kinds of infectious agents, such as fungal, bacterial, and viral pathogens, through the identification of their DNA in high-risk patients. For this, it is the most used nucleic acid amplification test. This is because of its much greater sensitivity, which outclassed everything else at the time of its creation. Other methods, such as histopathology and radiological imaging, are not disease-specific, while culture from samples takes a long time to grow and are not specific enough. PCR is considered effective because it is used on pathogens that are hard to culture in a way that is both sensitive and fast. Polymerase chain reaction limitations. However, PCR's sensitivity can actually be one of its biggest limitations. Wassenegger states that even the slightest contamination can serve as a template and thus can be amplified. This can cause undesired DNA to be replicated instead, often wasting time and money and skewing results. The samples for nucleic acid amplification must be good quality, and it also must be of a certain amount to be successfully copied. To successfully use PCR, you need to know information on the sequence you would like to replicate. This is because you need the right primers to amplify the exact sequence and thus copy the section of DNA you actually desire. This means that if you don't know what you're looking for, or simply don't know enough about the genes you want, PCR may not be the correct tool for your purpose.
Potential future applications or extensions of the technique. Since PCR was discovered in the early 80s, its use became routine to every labs to clone genes, diagnose disease, or identify genetic fingerprints. And many extensions of its techniques have been developed, and nowadays there are a significant number of them. PCR has yielded many generations of its methods and machines. So some of these extensions are endpoint PCR, multiplex PCRs, long PCR, quantitative, and digital PCR, to name a few. Quickly, we can have a look at this progress through these three techniques. For example, the endpoint PCR, it was the original version of the technique. It was created in 1983 and is still very useful in labs. However, only provide relative and qualitative results. The quantitative PCR, it was developed in 1992, is also popular, and this technique has enabled quantitative detection and also provides an absolute result. And finally, the digital PCR or dPCR, more recently, has also become readily available. And compared to conventional PCR technologies, dPCR enables a more precise, sensitive, and reproducible target quantification. This evolution in PCR technique that has happened and is still happening over the years is essentially to solve specific problems associated with the technique or to improve its performance. Some of the improvements that researchers are looking for are speed, for decrease the wait time for results. They're looking for continuous flow to improve the heating and the cooling system of the technique. And they also wish simplicity, making possible run PCR tests in simple low-power device and reducing costs of diagnostic tests. So it's possible to see potential applications of the PCR in areas with poor medical facilities. The lack of resources, such as electricity, clean water, or transportation, would not be a barrier for the use of this developed technique. Due to the evolution, it's possible to achieve the sample preparation, amplification, and interpretation of the results done in the same device. And this is the future of the PCR. As Chow and his colleagues said, PCR extensions have found widespread application in medical diagnosis, environmental monitoring, and food safety. Here we conclude this presentation. Hope you had enjoyed it. Next are our references on this topic, and thank you for watching.